Okay guys, so in this lecture we are going to talk about the regulation of transcription. In the previous lecture we talked about the regulation of gene expression. This is a continuation of the same lecture. Basically, regulation of transcription. The link to that lecture has been given in the description. So if you haven't checked that out, so please do check that out before viewing this lecture. Alright, so basically what is regulation of transcription? Regulation of transcription. Okay, in the previous lecture, we talked about regulation of gene expression. One way of regulating the gene expression is by regulating transcription. Transcription, as we know, is the first step of gene expression. It is controlled, it is the synthesis of an RNA molecule using a specific sequence of DNA called gene. This transcription is controlled by regulatory sequences of DNA. They are called cis-acting elements of the DNA, which are embedded in the non-coding regions of the genome. Interaction of these sequences with the regulatory molecules, such as the transcription factors, are transacting, which are transacting. They are called transacting, and we are going to talk about why they are called, the DNA sequence is called cis-acting, and why these transcription factors are called transacting in a bit. Okay, so the interaction of these regulatory molecules with the DNA sequences can help to regulate the transcription. They can either induce gene expression or they can express the expression of a particular gene. Since transcription is the initial step of the gene expression, this is what I told you just a moment ago. Okay, so transcription, since it is the first step of regulation, first step of gene expression, by if we regulate this step, we'll automatically regulate gene expression. And in fact, this is the most efficient way of regulating gene expression. And this is a way of regulating gene expression in both prokaryotic cells and eukaryotic cells. This is fully functional in both of them because transcription occurs in both prokaryotes and eukaryotes as well. Now, then we talked about the two components which are involved in regulation of transcription. One is the regulatory sequences of DNA, and the other are the transcription factors or the transacting regulatory molecules. These are actually proteins made by some other gene at some other part of the DNA. When they come and bind to these regulatory sequences of DNA, then a particular gene is either expressed or is repressed. Now, moving forward, specific regulator DNA sequences, the cis-acting elements basically, they are called cis-acting because they are usually located on the same chromosome as the genes which they are controlling, which they are expressing or which they are regulating. So these regulated DNA sequences, they are arranged on the same chromosome as the genes they are regulating. So that's why they are called cis-acting elements. Cis means on the same side or on the same part of the DNA. But the regulatory factors are called transacting because they are not on the same side, as they are not made on the same side as the genes on the same part of the DNA that has to be on the same side of the genes that they are controlling, that they are regulating, but rather they can transit from the gene where they are made to the regulatory sequence of a gene on another chromosome as well. Now, so basically in this paragraph, we talked about why the DNA sequences are called cis-acting and why the regulatory molecules are called transacting. Quick review, their DNA sequences are cis-acting because they are present, they are located on the same chromosome as the genes they are controlling, and the regulatory molecules are called transacting because they can transit from the gene where they are made to the regulatory sequence of a gene which they have to control or regulate. So the two components of regulation of transcription. Now, how do the regulatory factors bind to the DNA sequences? They bind to the DNA sequences through motifs. Okay, what are motifs? Motifs are basically some special super secondary structures. They are made up of two or more secondary structures of a protein. They are also proteins basically, but they are made up of two or more secondary structures of the protein which are arranged in a stable configuration. Example, the zinc finger, the leucine zipper, and helix turn helix. Okay, so these are basically the three types of motifs, three examples of motifs basically. Right, so I've made a diagram of zinc finger right over here on the right hand side. I've taken this from Lippincott Illustrated Reviews for Biochemistry, your textbook. It contains two antiparallel beta strands, right, and a helix, an alpha helix, and a beta turn as well. Okay, so several secondary structures are arranged in a stable configuration, and these secondary structures are coordinated with a central zinc atom through two histidine molecules and two cysteine molecules. Okay, so histidine amino acids and cysteine amino acids are coordinated with the zinc in the center. So, and this motif, the zinc finger, actually binds to the DNA regulatory sequences in order to bind the regulatory molecules to the regulatory sequences of the DNA. Moving forward, now, how does the regulation of prokaryotic genes expression work? Okay, so as I told you earlier in the previous lecture that prokaryotes can only regulate gene expression by regulating transcription. Okay, because in prokaryotes we know that post-transcriptional and post-translational modifications do not occur. So the only way of regulating gene expression is by the regulation of transcription. So now it is mainly regulated by controlling the transcription, the first step of gene expression in prokaryotes. It can be done by the initiation of transcription or by immature termination of mRNA being transcribed. Okay, so basically immature termination of mRNA being transcribed. This is one way of repressing the prokaryotic gene expression, and we are going to talk about this in detail in the upcoming lectures. And by initiation of transcription, we are going to talk about this as well in the upcoming lectures in great detail. But now let's move on forward. Now, mRNA transcription from bacterial operon. Okay, first of all, let's define what operon means. Operon is actually a single functioning unit of DNA, a complete functioning unit of DNA, and its components include a cluster of structural genes, basically two to three or maybe more structural genes, which are sequentially grouped together along with the cis-acting DNA elements, the regulatory sequence of DNA. These are promoter operators. Okay, we are going to talk about them, so don't worry. That regulate the expression of these genes is called an operon. Okay, so basically a DNA functioning unit containing a cluster of genes right over here and the regulatory sequence of the DNA. This regulatory sequence of the DNA regulates the expression of these genes. Okay, and the regulatory sequence usually contains these two parts, the promoter region, the operator region, and we are going to talk about both of them in detail. Okay, so the definition of operon, so basically, oh yeah, one thing, operons are only present in prokaryotes. Okay, they are not present in eukaryotes, they are only present in prokaryotes. And by the way, these genes which are clustered together in a single operon They are transcribed collectively onto the same mRNA molecule. They are transcribed collectively. All these three are transcribed collectively on the same mRNA molecule. This mRNA molecule, which contains more than one genes, okay, which contains the codons for more than one genes, is called polycistronic mRNA molecule. Okay, polycistronic. The cistron means a gene. Okay, so polycistronic means contain codons for multiple genes. Whereas in eukaryotes, since eukaryotes don't have operons, they do not contain a functioning unit that is controlled by the same operator and promoter. Actually, so in eukaryotes, since operon is missing, each gene is transcribed onto a single mRNA molecule. And this single mRNA molecule, since contains codons for only single gene, one mRNA molecule contains codon for one gene. That mRNA molecule is called monocistronic. Monocistronic mRNA molecules are present only in eukaryotes but not in prokaryotes, because prokaryotes contain operons, and the mRNA molecules which are made as a result of transcription from this operon is called polycistronic because it contains codons from multiple genes. Now, let's talk about the role of these two regulatory regions of the DNA, the regulatory regions of the cis-acting components, the cis-acting sequences of the DNA, the operator, sorry, the operator and the promoter. Now, okay, so let's talk about the role of the operator. Okay, in here I've talked about the role of the promoter as well. So let's get started. RNA polymerase, which is the enzyme that is responsible for the synthesis of RNA, which is responsible for the initiation of the process of transcription, it can bind to this promoter region. This promoter region, the role of this promoter region is that it is a point where the RNA polymerase binds. Once it binds, it initiates the transcription factor by passing over the operator site, and then it passes over the structural genes in order to transcribe them. If we look at this diagram over here, the RNA polymerase will come and bind at this promoter region. After binding, it will pass over the operator and then will go all the way over these genes, transcribing each of them, forming a single polycistronic mRNA molecule. But if this operator, now it's the role of the operator comes. If this operator is bound to a repressor protein, we've talked about the repressor in the previous lecture, that it is basically a regulatory molecule that prevents the expression of a particular gene. So if the operator is bound by a repressor protein, then the RNA polymerase is blocked. If this is bound by a repressor protein, then the RNA polymerase cannot pass over the operator. It is blocked. And if it is blocked, then what happens? The transcription will not occur. These genes will not be transcribed. So this operator has a key role in controlling the transcription of these genes. Then, if an inducer molecule is present, it will come and bind to the repressor molecule that has already bound to this operator. As a result of this inducer-repressor binding, the repressor molecule will change its shape. Conformational change will occur in the shape of repressor, due to which the repressor will not be able to remain attached to the operator site. And once the repressor molecule has left this operator, the RNA polymerase will be able to pass over the operator and then resume its activity of transcription. Okay, so basically operator has a key role in controlling the expression of the genes with which it is associated. So RNA polymerase activity can be hindered, can be initiated if the operator is attached to a repressor or is free. This is how it controls the gene expression. Moving forward. Okay, a very important note. I told you earlier that operons are not found in eukaryotic DNA, but they are only found in prokaryotic DNA. So that's why prokaryotic mRNA is usually polycistronic, whereas eukaryotic DNA is monocistronic. Polycistronic because it contains more than one codon for multiple genes, and monocistronic because contains only a single codon coding for a particular gene. Now, codons for only single gene. Now, moving forward, the mRNA which is transcribed from prokaryotes is called polycistronic mRNA, having codons from multiple genes that can be transcribed into multiple polypeptides. Whereas this one, the mRNA of eukaryotes is monocistronic because it is capable of coding for a single polypeptide only because it contains codons for only one polypeptide. Okay, so that's it for this lecture. And in the next lecture, we are going to talk about the Lac Operon, right? A special type of operon also present in prokaryotic cells. Okay, so thank you so much. The link to that lecture will be given in the description, so please do check that out. Thank you so much.